How about now?
Yeah, we can see it. Beautiful.
Okay, great. So in the interest of time, and because the session is already kind of finished, I'm going to skip a lot of interesting and important details. But if you want all of the details, you can scan this QR code and it will take you to our archive paper, which is much longer. So this work is a collaboration between Harrison Smith from the Earth Life Science Institute and Blue Marble, and me, Lana Snapayan, from Sony Computer Science Labs and the National Institute for Basic Biology. So I'm going to skip this, and I'm going to directly explain every word of the title of our paper, because I think a lot of A-Life people are not familiar with astrobiology, and there could be more links between these two fields. So the first thing is planetary scale. What I mean is that the unit that we consider is the size of a planet. And then there is replication, which makes it sound like we take one planet and then we get two planets. It's not exactly that, but it's kind of similar in spirit. Then agnostic. So this is the part that is probably most of interest to A-Life researchers. So here by agnostic, we mean that we're looking for something that is independent of the biochemistry of a specific type of life. So life as it could be, which is the heart of A-Life. So we're proposing a planetary scale replication. We're proposing planetary scale replication as an agnostic biosignature. And the biosignature is a clue that you can use to try to detect if there is life on a planet that is outside of our solar system. And everything we did in this work is in simulation. We haven't used real data yet. Very important precision. So our assumption in this simulation, we have three assumptions. The first is that life exists in the universe outside of Earth, and we're trying to detect this life. Second assumption is panspermia, the idea that life can travel through the universe. We know that this is possible because we humans have inadvertently sent bacteria to the moon. And we also have found that things like tardigrades can survive in space, so panspermia is possible. And the last assumption is terraformation, which in this case really simply means that when you have life somewhere, this life will change the environment. So for example, when you have plants, the plants will emit oxygen and change the composition of the atmosphere. But you can also think about this guy who, intentionally sends rockets somewhere and then tries to maybe terraform another planet. So the questions that we have are, first, under which conditions can we detect terraformation? And second, if we detect terraformation, can we actually pick out the exact planets that are being terraformed? Just to make the point that these assumptions aren't completely out of the possibility. So in the universe, we have up to 2 trillion galaxies. And each of these galaxies may have, on average, 100 billion stars. And of course, around these stars, we may find one or several planets. So the universe is huge. There might be something out there. It wouldn't be super surprising. So the base idea of our model is this. You have a bunch of planets, and you are able to measure somehow the chemical composition of these planets. It could be the atmosphere, it could be something else. So you measure the composition of these planets, and let's say that you find that these 4 planets, which are suspiciously arranged in a straight line, all have 20% oxygen in their atmosphere. When you look at this, it feels like it could be suspicious. And what we want to measure is exactly how rare would this pattern be. So could you get that by random if you just have a lot of planets, or is it extremely unlikely to get this at random? And the measure that we use is called the Mantel coefficient, which is simply the correlation between the variations in the position of the planets and the chemical composition of the planet. And it's not just that, oh, we have similar planets all at the same position, therefore it's suspicious. It's really more that you want to see that if two planets are close in space, they're also close in composition. If they are far away in space, they're also far in composition. The basic idea being that if you have life on one planet and it moves to another planet, it might terraform that other planet so that the environment is more similar to the first planet. And this terraformation process will not be perfect, and the life will also be changed by this new environment. So instead of having 20% oxygen everywhere, you might have the second planet have 19% oxygen. And then the third planet would be maybe 15% or 25%. So you want to measure these variations. And because this measure can apply to any kind of composition, this biosignature would be completely agnostic to the type of life that might exist there. So this is our model. You have 1,000 planets. One of these planets has life on it. Life goes to a second planet and changes the composition of that second planet. Not completely, so you don't get exact replication, but you get some kind of replication with mutation. And then you repeat this process going from the second planet to a third planet and modifying it, etc. So we calculate the Mantel coefficient and also the p-value of this Mantel coefficient. And it is this p-value that will tell us how unlikely this pattern is to appear at random. So I don't have time to explain the details here, but the Mantel coefficient, to calculate it, you take the physical distance between each pair of planets, you put that into a matrix, and then you calculate the distance between the chemical composition of each pair of planets. And now we can calculate the correlation between these two matrices. If the correlation is high, you get a high Mantel coefficient. If there is no correlation, then you get a low Mantel coefficient. Then you can take one of the matrices, shuffle the values at random, and recalculate the Mantel coefficient. Do that 1,000 times, and it tells you how unlikely your first value was. So you want to see a low p-value. So this is one example of a rod with specific parameters. So the x-axis here is the percentage of terraformed planets. So it's kind of equivalent to time. In black, you have the value of the Mantel coefficient, which, as expected, goes up to a certain point. And then in red, you have the p-value, which gets low pretty fast, much faster than the Mantel increases. In this example, we have a p-value of 0.01 at about 7% of terraformed space. We did this for a bunch of different parameters, so you can see everything in our paper. And so the answer to our first question is, yes, we can detect terraformation with high confidence. In this case, at 7% of terraformed planets, we have very low p-values. But this only tells us that terraformation is happening somewhere. It doesn't actually tell us where terraformation is happening. So we need a different pipeline for this detection. The pipeline that we use goes like this. First, we cluster the planets by composition. So we have groups of planets with similar composition. And then for each of these clusters, we calculate two values that are independent from each other. The first value is how much this cluster is contributing to the value of the Mantel. So if it contributes nothing, then it's unlikely that this cluster is the terraformed planet. If it contributes a lot, then this cluster is suspicious. Independently of that, we also calculate how spread in space this cluster is, with the idea that the universe is huge. So if some type of life was terraforming stuff, it wouldn't be in the whole universe. It would be confined to a region of space. And so if we have one cluster of planetary compositions that contributes a lot to the mental value and is very confined in space, we consider that a detection. So here is an example of detection. You have 1,000 planets. This is in 2D, but projected this is in 3D, projected in 2D. And here, all of the terraform planets are in blue, and the detected planets have a red outline. So here we have 40 planets that are terraformed. That's 4% of the space, and we detect about half of them. And this is the lowest value at which we have a detection. So this figure is just to show that our detection method is highly specific, and by that means that we basically don't have false positives. We might not detect all of the planets, but at least when we have a detection event, it's usually accurate. It's usually true. So future work, we want to apply this method to real data. NASA has a public catalog of exoplanets with more than 4,000 planets detected for now. We have a few issues that we have to work on, like the initial distribution of planets is likely to affect the results. Even more important than that, planets move a lot relative to each other, so you get some kind of space mixing, and that will definitely affect the results a lot. Finally, if you haven't seen this series or if you haven't read the book, I can highly recommend it. The main thing is that planetary civilization in space are trying to hide from each other. And my point is that with our method, if you don't really want to hide, you shouldn't go around terraforming planets. And finally, I want to thank my co-author, Harrison Smith, people who discussed with us and reviewed the draft, so Cole Mathis, Estelle Jana, and Stuart Bartlett, and you for staying after the time of the session and listening to my talk. Thank you.